think we're gonna get started, y'all. Welcome to the next session. My name is Katie Thompson-Peer. I'm an assistant professor at the University of California, Irvine, and it's my pleasure to introduce our next speaker. Our next speaker conducted his PhD research at Stanford in the lab of Tony Wiscori. And then instead of doing a postdoc, he jumped right into running his own research lab as a Sandler Fellow at UCSF, at the University of California in San Francisco in 2012. Since then, he's been promoted to assistant professor and then associate professor, and is the associate director of the Bakar Aging Institute at UCSF. I was a postdoc at UCSF for a number of those years. Our paths didn't overlap a ton, but I do remember seeing a video or a picture of him in one of the department holiday lab parties where he was in a costume stitched to another professor as two mice experiencing parabiosis, much to the joy and laughter of the rest of the department. I also remember him speaking very passionately about our individual and collective responsibilities when it comes to promoting equity and inclusion in academic science as part of a panel discussion for a student group that was called Students for Diversity at UCSF. So I eventually left UCSF and came to UC Irvine, and I run a research lab here at UC Irvine. And in addition to running a research lab, I also teach a class. And the class that I teach is the first class that incoming freshmen take in biology when they come to the UCI. So it's their first exposure to college-level biology. And so as part of this class, in addition to teaching them things from the textbook, I also want to introduce them to literature, to the scientific method, to reading papers in journals. And so for one lecture, I dedicate this to telling them, how do you look at a journal article? How do you understand the graphs? How do you kind of pull it apart? And for that lecture, I need a journal article that is both so exciting that the freshmen who are just new to biology are going to get excited about it, and also clear and easy for them to understand. It has to have graphs of all different kinds, bar graphs and scatter plots and line graphs and all that, and it has to be beautifully diagrammed and labeled so that the students can understand it. And the reason that I'm bringing this up is that for the past 2 years, I have been using one of Dr. Villeda's papers for my Intro to Biology class. It's his 2020 publication from Science, and I use it because not only is the story elegantly explained, but the figures are just gorgeous and easy to understand and easy for undergraduates who are new to science to figure out, what are all these bar graphs doing and what am I trying to learn from this? And so that is my challenge to you today, and the bar that I try to aspire to in my science is to do not only science that is exciting enough that new freshmen can understand it and it can get published in top-tier journals like Science, but also that is clear and easy to understand enough that people aren't afraid of the science but can get excited about it. And so I really that's part of why I'm so inspired by Dr. Villeda, and I look forward to listening to hear him tell us more today. So with that, please help me welcome Dr. Villeda to the stage.
Hi, everyone. Good afternoon. I just got really nervous after that introduction. Thank you so much, Katie. That's crazy. Just fair warning, they gave me this chicken, and I really want to use it somehow throughout the talk or in the questions. Also, thank you so much to Mike and Manuel for the invitation. This is a real treat for me. Usually I tend to go more to aging and biology of aging meetings, so just being immersed in sort of cognition and learning and memory has been really great. So today I want to focus a little bit on this idea of what we call rejuvenation. It's a bit of a controversial word, but really in essence what we're trying to do is really just ask about reversibility. Now let's see if I can do. So the big question in my lab is sort of this idea of reversibility. So not necessarily just can we slow down aging, but rather can you take an already sort of aged organism and can we actually bring back function? Now the way that we're kind of defining rejuvenation is from a functional perspective. So can we function closer to what we were functioning as during youth? So the way we think about this is, I'm a neuroscientist by training, and we think about cognition a lot. And one thing that we know is that when you're young, even middle-aged, there is an overt sort of decline that's happening. There is some decline, but it's not really overt. But it's really around that time where you kind of retire, where you start noticing that cognitive decline. Now, along with the sort of cognitive decline that we're seeing, we also have like an increase in sort of the vulnerability towards neurodegenerative diseases. So right around that same time, as we're sort of seeing this decline, we're also seeing this disease. But what's interesting, even in things like Alzheimer's disease, if you're 20, even with genetic predispositions, you don't have that overt pathology. So something naturally is occurring when you're young that's able to stave off that aging or age-related decline. And that's what we're really interested in kind of understanding, which is really what are those mechanisms of youth? And basically, as we're trying to leverage this sort of biology of aging, we really are thinking about it in terms of restoring function, and in doing so, increasing what we call health span. So we're not changing lifespan, we're not trying to tackle how long you live, but rather within some preset amount of time, we just want to shrink the amount of time that you're actually in disease state and increase the amount without disease. And we think that by studying basic biological mechanisms of aging, we can actually tackle on a number of different diseases, from mild cognitive impairment to Alzheimer's, without necessarily targeting any one specific disease. And the way we focus on my lab is on interventions. We're trying to figure out both mechanisms that are changing as we get older and interventions that could re-engage those mechanisms, potentially improving function. So what kind of function do we look at? Well, in my lab, we focus on very stereotypical decline, sort of well-conserved sort of learning and memory tasks, ones that are also conserved in their impairment with time. So one thing we focus on is spatial learning and memory. Here, I'm from San Francisco, so we're just going to have a parking lot. This is just spatial learning and memory, which we hear about. And basically, we're just going to use the cues around you to find your car. So in this particular case, I have sort of this red Tesla, and I parked it closer to the Golden Gate Bridge, a little bit further away from the rainbow flag. No problem when you're young, but then you get old. And then all of a sudden, you're going to be in that parking lot with your car alarm running around for like 15 minutes before you actually find it. So that's your brain on aging. Full disclosure, if you're not like an octogenarian, it's also your brain on chronic stress, but I kind of give that now because people used to freak out that they were aging too fast. Chronic stress or aging cause both of these. So in my lab, we use animal models so that we can actually then manipulate and understand from a mechanistic standpoint what's happening. And we use the mouse model, and it can actually recapitulate a lot of different features of aging. Here I'm showing you a young animal around 4 months of age. That's roughly the equivalent of sort of a human in their 20s. And I'm showing you close to a 2-year-old mouse, and that's roughly someone closer to 70. And as you can see, a lot of features we see in ourselves, for example, this older guy getting a little bit more plump, the gray hair, all of that are signs of aging that are recapitulated across species. Now, likewise, in the brain, we also have conserved age-related cognitive decline. Now, in particular, going with that spatial learning and memory, this provides us really nice models where we see similar decline and simple tasks where we can start asking what promotes decline, what brings back function. So similar to finding your Tesla near a bridge, you can actually then teach these animals to find a hidden platform just in relation to these spatial cues, in this case, closer to a triangle. Now, an older animal can find the hidden platform just like a younger animal can, but they make more mistakes. In this particular case, they go in the wrong direction multiple times. So in the context of normal aging, not with any sort of genetic manipulations, we can start asking what's promoting it, what can potentially reverse it. And that's really what sort of we're focused on. Now, this is governed by one part of the brain, the hippocampus. This is actually the most susceptible region. It's the first point of contact between aging and your brain. It's in the hippocampus. Now, with aging, there's a really rich body of literature that's really defined a number of cellular hallmarks of aging. Now, with aging, in the absence of disease, we don't have gross cell death, but rather what we have are really cellular and molecular changes that are happening. For example, regeneration, things like stem cells. You have inflammation with things like microglia or other glia that become reactive. Your vasculature, your blood-brain barrier also changes. It actually changes in what can get in, the cell activity. Eventually, of course, that's going to impact the neurons and the way that they can actually communicate and synaptic plasticity. The stability of the dendritic spines is changing. It's much more dynamic, less stable. And then collectively, that leads to cognitive impairments. I will say that each one of these is on a different trajectory, and each one of us in this entire room is aging at a slightly different trajectory. So for some of us, our drivers might be regeneration. For some of you, it might be inflammation. For others, it may be a leaky blood-brain barrier. The idea is understanding, well, how does each one change? What's the combination of cells that we actually have to target in order to improve cognition? That also means that it may be different across the different individuals. Now, in thinking about aging, I'm a neuroscientist, but we think about more than just our body. And this is where this model of parabiosis came in that Katie kind of mentioned. And yes, I did dress up and attach myself to a more senior faculty, and we had a whole moment. I was like 30-something at the time, so forgive me. But what we're doing here is basically we're taking two animals, and then we're connecting their vasculature together such that two hearts pump one bloodstream, and we can mix young and old blood. And the reason we did this, people often ask me, well, why are you looking outside of the brain if you're interested in the brain? Well, with aging, everything ages. We see this in the mirror. You're getting gray hair, you're getting wrinkles, and you're getting cognitive decline. So we can't ignore the interface, the interactions between the brain and the rest of the body. So that's what we're looking at, is can we learn something from the periphery to understand the brain? And what we found is two things. Number one, old blood's pretty bad, and we have pro-aging factors, most of them immune in nature, so inflammation in your aging immune system is a driver of cognitive decline. And the opposite is also true. Young factors are also sort of potential to reverse sort of aging hallmarks. But it's not just about parabiosis. Whoa, can we go back? I don't think I'm in control anymore. Okay, thank you. So it's not just about parabiosis and physically connecting these animals, but rather we think it's about factors potentially that are in there that are signaling your brain potentially. So we saw parabiosis can reverse things like plasticity and neurogenesis, but we can't do behavior in mice that are connected. People in my lab did try it, and I don't recommend it, especially in a pool of water. So we had to come up with an alternative to having two connected animals there. And what we did is we just inject blood plasma. Basically, we remove most of the cells, not all of them, but most of them, and we can just inject it over a month, and these animals can actually perform better. So I'll show you some of this primary literature. Here, we're just looking at number of errors. So variation on the Morris water maze, we divide it into a pool of water, and then we can just count number of errors. In this particular case on the right, the animal made 3 errors before finding the target. And then we can quantify errors. That gets around the young versus old, the faster versus slower. It becomes much more quantifiable that way. So in gray is just an old animal that received old blood, and they're doing pretty poorly. By the end, they're still making over 2 errors. But when you give this young sort of blood plasma, it actually was much improved. It was closer to 1 error. So that suggests there really is factors in blood. And number 2, you can improve function. You can actually make an old brain function as if it were younger. So we were really excited about this. And for a long time when I started my lab, I thought young blood is it. This is so unique. It's super special. And of course, I'm wrong. And the question and the topic is much broader than that. So it's not just about sewing mice together, but rather there's a number of different systemic or lifestyle interventions, things that affect the entire organism that are known to improve function, things like caloric restriction and exercise. And my lab's really interested in understanding, well, is it something special about young blood factors, or is it more fundamental than that? Is it that your peripheral organs or tissues are communicating with your brain? Perhaps we have messages that say, hey, you just exercise, boost your memory. Or maybe it says, okay, you're feeling younger, boost your memory. And we're trying to sort of decipher what that language is, what those signals are within the blood that can promote this function. So I'm going to tell you 2 vignettes, one of them on the young blood aspect and one of them on exercise, a little bit of kind of where we are right now with the paper that we published a few years ago. So the question that my postdocs were really, really interested in, and this is work by Adam and Britt, really talented senior postdocs in the lab, they're really interested in asking, well, what are the cellular and molecular mechanisms then by which this young plasma can even rejuvenate? I will mention this, the FDA has come out and said we cannot just do blood transfusions into the elderly to try and treat Alzheimer's. Number one, there's not enough plasma in the world for the people that have it now, let alone in 50 years when this tsunami goes crazy in terms of the people that are getting older. But really, we need something that can actually be sort of defined, prescribed, regulated, and treated. And that's where the idea of how does it work comes into play. We really need to understand the mechanisms behind it. So the first thing we did is we just took a step back, went back to that plasma, and just asked, well, what's the difference between young and old plasma in the first place? And for that, we used a proteomic approach. So in this particular case, just mass spectrometry. And we just asked, what's changing? And there were certain proteins that both went up and down between the young and old blood plasma here. And then we took all of the proteins in blood that were changing, and we just asked, well, are they involved in particular processes? Maybe the biology of what those factors are involved in can guide us in terms of how it's working. A lot of immune things came into play, but we also, as our top sort of biological process, came upon platelets, and those are sort of these kind of forgotten little immune cells. And we thought, okay, that's really interesting. And then when we went into our plasma, it turns out that we get rid of most cells, but it's actually rich in platelets. And perhaps some of you have heard about platelet-rich plasma. Kobe Bryant, when he was still alive, treated it for his knee while he was a basketball player. It's very controversial, so we're like, oh boy, platelets. Okay, let's see what this has to do with young blood. So this time we focus specifically on just these little cells. They're enucleated, they don't even have a nucleus, but they're involved in things like wound healing and coagulation and immune responses. So we now took those platelets, or that fraction that has those platelets, and did proteomics as well. And we wanted to know, well, if we zoom in, we went from parabiosis blood now to plasma to platelets, and then within the platelets, then what's changing? We really like to take that reductionist approach. Now, we got a number of factors, and we wanted to know, well, have any of them ever been even associated with any other intervention? And we were really fortunate in that there was one factor that actually had been associated. It came up in a lot of different proteomic analysis, and it's been implicated in exercising young animals, and it's been implicated in this other kind of weird rejuvenation approach where they take half of the blood of an old animal and then replace it basically with saline, and that also can have an effect on it. But the one factor that keeps coming up across all of this is this factor, PF4. It's secreted by the platelets. So the first thing we wanted to know is just, okay, proteomics, we have to validate what's going on. So we went into an independent cohort, and here I'm just showing you a Western. You can see really, really bright signal on the left and very dim signal on the right. That's just the platelets really do have a lot more when they're younger. We like to focus on factors that are conserved across both humans and mice. So we actually recruited a small cohort of humans that were between either 20 to 34 or above 61. Most of them were in their 70s. And likewise, in the blood of those elderly humans, we also saw that it was higher in the young versus the old. And that's how we kind of decide what factors to pursue, is which ones are also changing in these humans. So what exactly is this factor that I'm talking about? So platelet factor 4 is known as a chemokine. It's an immune molecule. And what it does is it gets secreted by these little platelets, it goes into circulation, and then it can bind onto different receptors. And depending on which receptors it binds to, it can affect a number of different things. Like I mentioned, wound healing, blood coagulation, immune function. So really it depends on kind of who it's interacting with. And we thought that was kind of interesting. So the question we wanted to know is, all right, can increasing this one factor, can it recapitulate at least some aspects of blood? Can we actually promote some of this benefit, not necessarily with the platelets or the blood, but now really these recombinant proteins, because eventually that's going to get us much, much closer to the human condition. And what we do is we inject it over about a month, and then we do behavior. I'm going to show you for this talk, I'm going to focus mostly on that water maze that I described with the young blood. Now, what we saw is because it was an immune factor, we wanted to go through some of the hallmarks of aging. We first started with the immune hallmark, which is neuroinflammation. So here I'm just showing you a stain in red of microglia, so the resident macrophages. And in green, I'm showing you when they get activated, they kind of aggregate this protein known as CD68. That kind of tells you that they're reactive in some way. When you're old, it's just bad. Inflammation is really high. When you give young blood, that goes down. And when you give platelet factor 4, we actually saw the green, that reactive marker, actually go down. We also wanted to take a broader approach, like a more unbiased approach. So we actually just did RNA sequencing of the whole hippocampus and just asked, transcriptionally, gene-wise, like what genes are changing? And it was a lot of genes that changed, close to, I think, 900. And then when we looked at, well, what exactly are they related to? A vast majority of the genes that went up following platelet factor 4 had to do with synaptic functions, very specifically. So even though we're taking the whole hippocampus, every cell that's in there, the genetics is telling us that's really having a change at the synaptic level, really the neurons. So that got us excited. And then when we looked then at the actual behavior, what we see here is, on the first day, there's no difference. But when you start going on the second day, the testing day, the animals actually continued to perform better, particularly as the task continued. I will mention this is not the same magnitude as young blood plasma. So every time you go down from the parabiosis to the plasma then to the factors, you recapitulate a fraction of the effect. So again, we think this is really indicating multifactorial, probably additive mechanisms, but one factor was sufficient to give us functional improvement. So then mechanistically, we want to know, how is it working? I love this idea of like inner organ communication. So we now know, okay, platelets must be talking, or your immune system must be talking to your brain. How? It's either going to go directly into the brain and potentially signal, or it's going to go through some part of intermediary, some middleman that's going to have some function. So the way that we did is we used a trick, and we made a form of this factor, play factor 4, that we can actually track. And the way we can track it is anytime, wherever it's present, we actually have a reaction that causes luminescent signal. So we put this into first the liver, it went into the blood, and we just asked, what tissues can we actually sort of see this factor going into? Now here you see bioluminescent signal, and it's really, really high in blood. It's pretty high in liver, but when we looked across the different brain regions, we just couldn't detect it above sort of that control level. So that means it's not going into your brain. It must be working through some middleman. Something outside of the brain is signaling. So that suggests, okay, why don't we go back then into blood? So let's try and figure out something in the brain by looking again at the blood. And we just took an unbiased approach. We went back to that sort of proteomics approach where we just look at all the factors in the blood, and we just ask, well, what's changing then in the context of this factor? And it was a lot of immune responses. Everything was suppressed. We kind of saw a dampening of inflammation in the periphery. And we're like, all right, we have a player, a candidate. So then why don't we try and look at then the immune system itself? We wanted to know both what cell types might be changing, but also transcriptionally what's changing. Is the immune system, changing either cell type or molecular-wise? We use single-cell RNA sequencing. All you need to know is that we're able to detect every part of the immune system, both the number of cells as well as what's changing within them with respect to gene expression. So here, if you look at the young and old graph here, all I'm showing you is very broadly. As you get older, your immune system changes. There's a big shift in the types of cells that you have. And that shift actually went back closer to what it looks like in a young immune system when we gave this factor, suggesting that really you can rejuvenate even the peripheral immune system. Here I'm just showing you one type of cells. These are your T cells, and they've been really, really kind of gaining a lot of traction with Alzheimer's disease. You can see that between the young and the old, a lot of genes change. There's this entire profile that emerges. But when you sort of expose them to some of these young blood factors, you can kind of go back a little in part. You're partially kind of reprogramming them back towards that youthful state. So we got pretty excited about that because we're like, okay, we now have a factor that's completely changing the peripheral immune system. We know inflammation is a driver of disease and cognitive decline. So could this potentially then be having a benefit by impacting the peripheral immune system? And the way we wanted to tackle that is figuring out who are you talking to. Now, what we did next is we said, okay, there's a lot of different receptors and sort of things that this can communicate with. And we looked at all of them and asked, are there any that are expressed in the peripheral immune system but not in the brain? We really wanted to focus on ones that were just outside of the brain. And we looked at publicly available single-cell RNA sequencing, our own single-cell RNA sequencing, and we found one receptor that was really not expressed in the hippocampus almost at all, but it was expressed a lot on cells like those T cells. So we thought, okay, let's see whether potentially this young blood is targeting then subsets of the peripheral immune system to have a functional consequence on the brain. So in this particular case, the postdoc took a leap of faith and aged a knockout animal that's missing this receptor for 2 years and just said, well, let's see if this is going to work. We do a lot of this in my lab. I don't know how we convince them to do it, but people do this all the time. So we have this knockout. It's 2 years old, no longer expressing that receptor on the immune system. It's predominantly on the immune system. And then we just add that factor, and we go, is the benefit still there or not? Now, when we looked at inflammation in our control, just like we saw before, there's a reduction in inflammation in those microglia. However, in the knockout condition, you'll notice that play factor 4 really doesn't do as much. So this is not everything. It goes through other receptors as well, but it does have a really strong effect at blunting it. When we looked transcriptionally at just the hippocampus, and we looked at all the genes that go up with PF4, but no longer go up in that sort of animal lacking the receptor, again, involved a lot in synaptic processes, really making a really direct link to sort of neuronal function. And of course, we like behavior, so we did behavior, and then what we saw here is consistent with what we saw. If you look at the solid lines, that factor improves it, and you have less errors. And then if you look by block 10, that's the best place where you can actually see it. The knockout is not as good. This is not a full blunting or mitigation, but it does suggest that in part, the peripheral immune system is doing something with respect to this sort of rejuvenating effect. And let's see if this works. There we go. So kind of putting this first part together, we're really interested in inter-organ communication, understanding how to bring back function in the brain by not necessarily even targeting the brain, but rather the rest of the body. We think we identified a really interesting role for these little cells, platelets. We think that they're impacting the peripheral immune system, dampening inflammation both in the periphery. That leads to decreased inflammation in the brain, and somehow that's now promoting function. Obviously, a lot more work, obviously a lot of black boxes, but we're really excited that we're at least starting to put together the pieces. But I mentioned it's not just about young blood. Other interventions can, so I'll quickly share a little bit about where we are with the exercise project. Now, I'm going to give you this summary. It was done in mice. I'm going to use this human schematic because I think it's adorable, so just go with me, but remember the findings were in mice. So we have these elderly organisms, and the question that my postdoc and graduate student had is, well, we know exercise is amazing even for people with Alzheimer's disease, right? It really is beneficial cognitively, but they're the ones that are frail. They cannot physically do the exercise, which would actually help them. Can we come up with an alternative? And the idea is, can we potentially transfer the benefits of that physical exercise through blood? Is there some signal in our blood that can tell our brain, boost memory in an exercise-dependent manner? Now, what we showed in animals, in mice, is that indeed you can take an old animal, and that's really important. This is not young blood. We're using old animals. You exercise them. You collect their plasma. You inject it to a completely sedentary old animal that's never exercised. You test them, and you get cognitive improvement. Exercise actually works even greater than young blood. So again, a lot of different magnitudes, a lot of different mechanisms involved. So going back to our animal, we wanted to find those factors, those individual factors. So we used that proteomic approach, similar to what we did with young blood. And I'm going to skip over a lot of this because it is published. We focused on one due to, again, proteomic analysis. It's called GPLD1. The first thing we always do is confirm, and you'll notice it's lower in sedentary mice than in mice that had access to this running wheel. We pursue factors that also change in humans, so we had access through elderly individuals over the age of 70 that had worn Fitbits, and we were able to collect their blood. And we segregated it based on activity, and similar to the mice, we also saw that elderly individuals that were more sedentary had lower levels, those that were active had higher levels of this factor. So we needed to know where it's coming from. And in our analysis, as well as in human analysis, it's predominantly from the liver. So exercise, when we looked at all the tissue, selectively increases expression in the liver, and your liver then secretes it into circulation. So what is this GPLD1 that I keep talking about? Well, it's an enzyme. Basically, it's a scissor. What it does is you have proteins inside of your cells, and they get modified so that they have a little fat tag called a GPI anchored. And what that fat tag does is it basically just anchors a protein to the cell surface. What GPLD1 comes is it acts as a scissor, it recognizes that fat tag, and it sort of releases those proteins from the cell surface. Now, when it does that, what can happen is those other proteins can go on and activate signals, it can inhibit signals, or the protein itself can just be degraded, and you no longer have it. So we were excited because that's a factor that can do multiple things, and we thought that's really important. So the next thing we wanted to know is just sort of ask, well, what happens then if we increase this particular factor? But we wanted to mimic biology, so we wanted to increase it in the liver and have the liver secrete it into the bloodstream. So we did that using a genetic trick where you just get the liver to express it. But we did one more group, and what we did is we made a form of that factor that can no longer have that cutting ability. It basically no longer recognizes those proteins to cut them. We also expressed it in the liver and in the blood. Now, what we saw when we then did behavior on these animals, let's see if we can, there we go. Those that had this, this liver sort of enzyme out in their blood, they performed much, much better. And you'll see that the different factors and the different conditions and the different interventions have different magnitudes. This is one of the stronger ones that we've seen. Now, when you prevent that enzyme from cutting, all of the effect goes away. Now, that's really important because now we start knowing, okay, I think we're figuring out how this might be working. It has to communicate then with cells through some of these proteins that are kind of tagged onto the cell surface. So this is where a postdoc in my lab, Gregor Bieri, came in, and he's like, okay, I want to know, what is it talking to? What are the targets and the cell types, and how is this being mediated? So the first thing that he did is he leveraged these publicly available data sets and just said, well, there's about 150 potential proteins that are tagged. Where are they expressed? And about, the majority were either immune cells. The immune system keeps coming up for us in aging, but it was also on epi and endothelial cells. And what that suggested to us is potentially it could be brain vasculature. Maybe brain vasculature is a target for our liver enzyme. And that would make sense in terms of that communication between organs. So what he then did is he wanted to know, well, is there some inkling that we can find that there might be an association there? So we went back to our human data. So we took those individuals whose factor we had measured, but we also had MRI metrics for it. And we focused specifically on white matter hyperintensity volumes, which is sort of a metric for sort of vascular microlesions, such that the larger the volume, the less integrity of sort of the cerebral vasculature. And what we saw is that in these individuals, the higher the level in the blood of this liver protein, the lower the volume of these white matter hyperintensities, again, suggesting an association between levels and vasculature, but enough to really encourage us to continue pursuing this potential connection. So what we then decided to do is really look sort of at the vasculature and ask, are there any of these potential proteins that have that fat tag that are on the blood vessels that maybe are implicated in blood-brain barrier? We got really lucky. There's a new Sandler fellow at UCSF right down the hall that's an expert on one of them, known as ALPL. It's really important for blood-brain barrier integrity. It goes up when you're older, totally changes the dynamics of your blood-brain barrier. The cell activity totally changes. So then we wanted to know, could this be one of our targets? So what Greg then did is he went in vitro, and he just said, okay, if I put together GPLD1 and I put together this enzyme on the vasculature, does it get cut? If it gets cut, I should be able to see it in sort of the media just floating around. And that's what he did. And what he saw is with GPLD1, it's floating in the media, it gets cut. But if you take away the ability of GPLD1 to cleave, it's no longer there. So it really is a bona fide target. He went now into the hippocampus, and he looked at the vasculature. And in red, you can see the vasculature of the brain decorated with this protein, and it goes up with age. The other thing is that it's not just about how much the protein there is, it's really about its activity. The activity of this protein increases and changes the blood-brain barrier. And we saw this increase with age. But then when we looked at our animals that had that liver protein and higher levels in their blood, we saw less expression on the vasculature and lower activity. And that got us really excited because we asked, well, what happens then if we mimic that inhibition? So no longer increasing this protein, but we just target the vasculature itself. And we did that by actually finding an inhibitor that we could infuse into food because we're trying to get a little bit more translational each step along the way. So these animals just eat this inhibitor, and then we can do behavior. And then what we saw is, in an independent cohort, this is now by Greg, you can see GPLD1 is really, really beneficial. But then when we target the vasculature and prevent that blood-brain barrier change that occurs with aging, we actually phenocopy. We actually get that same sort of benefit. Now, what that suggests to us, sort of putting this together, is really that in exercise, there's this liver-to-brain rejuvenation axis. This liver enzyme does not change with age at all, so it's not a young blood factor. And liver changes, actually, we don't see those in the young blood treated. So it really suggests that the different interventions have different mechanisms, different organs are informing your brain to boost memory. I think that'll be really important, right, as we start thinking about, can the effects of aging be reversed? Because each one of us is aging in a slightly different way. And it may come down to exercise factors may be great for people with blood-brain barrier or sort of lack of integrity, but other people may need those anti-inflammatory factors. So we're really trying to tease apart these different mechanisms to be able to give the best chance across the different sort of aging modalities. And with that, I'll just thank my lab and all the people that I try to thank along the way, as well as their funding sources. And with that, I'll take questions.